Okay, so let's talk a little bit about membrane functions and what membranes do. First thing we should talk about, I don't have a slide for this, but you should be thinking about this, is what things can cross membranes. Membranes are made up of bilayers of phospholipids interacting, one layer, one leaflet they call it, of phospholipids and their hydrophobic tails interacting with another leaflet on the other side, the other part of the bilayer, with their hydrophobic tails. So they make a separation that's hydrophobic components in the center there that separates hydrophilic pieces and separate inside aqueous water-based solvent areas from inside water-based solvent areas, right? So there's a barrier here, right? And we should be thinking about what can cross a hydrophobic barrier like this. And your reading should tell you this, but it should make some sense. Things that are charged or have a lot of hydrophilicity, a lot of hydrophilic component, will not cross. So amazingly, a membrane will not allow even a single proton that's positively charged to cross this membrane. You can actually separate protons, different concentrations across the membrane. So I have a lot of protons out here and no protons in here, and the membrane will keep those separated. The protons cannot even diffuse across a membrane, even though they're they're a particle essentially, right? A positively charged particle. So that also means that larger atoms that are charged will not cross, right? For example, sodium ion or potassium ion or chloride ions will not cross. Then that also means that other things that are charged, like amino acids, which have Zwitter ion charge on them, right, will not cross. So membranes are really good at separating charged things. What about hydrophilic things? Well, even there, small hydrophilic things will not cross. For example, water itself will not cross a membrane, right? You can separate water from other vats of water from the inside, from the outside, right? You need a protein to actually allow that water to cross. That means that other hydrophilic things, hydrophilic amino acids, how about glucose, which is even bigger, will not cross. Those things are too hydrophilic and not hydrophobic enough to cross the membrane, right? Proteins generally will not cross because they tend to have hydrophilic components, although if you have a really hydrophobic protein, it might actually be able to cross through the membrane. Now, what does cross? Well, things that aren't charged, like oxygen, for example. Oxygen is O2, there's no charge and no hydrophilic component. They will cross the membrane very easily, slip right through. CO2 will go through. This is the basis of how gas exchange happens in respiration in animals. Bringing oxygen into the body, into the blood to interact with hemoglobin, for one, and for two, oxygen crossing membranes to get into the cells and carry out cell respiration. The opposite is true for CO2, right? CO2 is produced through cell respiration. It will leave the cell easily through the membrane, go into a cell that's carrying hemoglobin, interact with hemoglobin, or be transported by carbonate, and then back to the lungs, where it will cross through plasma membranes of cells to get out into the air in the lungs. Okay, so let's talk a little about diffusion, important principle of how membranes work, right? Diffusion is essentially just a random motion of particles in liquid, gas, or solids. We're going to talk about liquid mostly in this case. Particles tend to diffuse from high concentration to low concentration until concentrations are equal throughout the medium, right? So that's a thermodynamic thing. Particles tend to want to equilibrate so that they're both sides of a barrier, there are equal concentrations. Otherwise, there's essentially energy stored in this. So this state here, with all these particles on one side of the membrane and none on the other side, is a high energy state. These particles want to go down their concentration gradient to become, to equalize the concentration. So they want to do this, right? If you make this membrane permeable to these particles, then they will diffuse until they're equal concentration. So this is an equilibrium state here, right? If this membrane's permeable. So if these are hydrophobic components or let's say oxygen or CO2, this will happen across the membrane, right? Osmosis is what happens if you have a barrier, semi-permeable barrier like this one, right, that doesn't allow molecules to cross, big molecules to cross, but does allow water to cross. So let's say you have glucose in here, for example. In glucose, you've put in high concentration on one side and low concentration at the other side across the semi-permeable barrier in between. Then water can cross, but the glucose cannot. So instead of diffusion powering glucose movement until the glucose molecules are equally concentration on both sides of the barrier, instead what we have here is we have water moving. That's osmosis. So the water will go from low concentration, or low solute concentration, to high solute concentration in this direction until they're equal. So you see these two now. The water has moved from left to right here and increase the volume on the right side of this until the solute concentration is equal to what's on the left side, right? And so that's osmosis, right? And osmosis is important in cell function. I'll show you in a second, right? But it's the kind of the corollary of diffusion, right? Diffusion is movement of solute, it's the solvent, the solute itself, where osmosis is the movement of the solvent. And that depends on the properties of the membrane that's separating the two systems. Okay, so osmosis is important in cells. I told you already that cells, membranes cannot allow water to cross through. Water will not cross through membrane on its own. So this tells you something that cells can actually absorb or release water depending upon whether they're in an environment that's what's called hypertonic or hypotonic, which tells you something about the membranes that these cells have. These cells have proteins in them that allow water to cross the membrane. Those are called aquaporins. So this is not happening because the membranes are leaky to water. It happens because the membranes actually have proteins in them that allow water to cross for various physiological reasons. All right, so let's talk about hypertonicity and hypotonicity. If you take a solution that has, erase this. All right, so if you have a solution that has different concentrations of solute on two sides of the membrane, one concentration is going to be considered to be hypertonic to the other. So hyper means more, tonic means solute concentration. So more solute is hypertonic. Oops. So in this case, all right, so in this case, this is hypertonic, and this is the opposite of hypertonic, which is hypotonic, or less solute concentration. So there's always a comparison made between two states that are separated by some sort of barrier when we're talking about hyper and hypotonicity. So this is hypotonic, right? Hypotonic relative to this hypertonic region, right? So if the concentration of solute is equal between them, then they are isotonic. Iso meaning the same, so isotonic, isotonic, right? And this happened after osmosis got going, right? So if you take a cell and you put it into a solution, red blood cells, let's say, and you put into a solution that's isotonic, so the concentration of solute inside the cell is equal to the concentration of solvent. Outside the cells are fine. They look normal red blood cells, right? Here we go, normal red blood cells. So if the cells are put into a solution which is hypertonic to the cell, right? So more solute concentration than the cells have themselves, the water will tend to leak, crossing through aquaporins. So the water leaves and the cells become collapsed, right? They become shriveled and lose their water, and they'll lose their function as a result too. If you put cells into a solution that is hypotonic relative to the solvent concentration inside the cell, water will tend to come into the cell, crossing the aquaporins, and those cells will tend to break because they'll swell until the membrane integrity is lost, and then boom, they burst and the cells pop. This is why when you have an accident victim at an accident site, lost a lot of blood, you have to give them plasma. Plasma is basically the solvent component of blood, has all the solvent concentration, all the ions, glucose, and so on and so forth, that make up the solvent. That would be the same solvent concentration as blood cells themselves, right? That's plasma. If you give someone water, their cells will lyse or burst. It's called lysing when the cells burst, and that will be the death of those cells, and that will not help the person who's had the accident. So giving water to replace blood is not good. Plasma, however, will work because that will increase blood pressure but not burst the red blood cells that are in there. And even better is you give them blood that's in a solvent, obviously plasma, that gives back people blood cells, that they have those to work with also. Matching blood type, of course. Okay, this happens in plant cells too. You see this all the time if you grow plants, right? So if you water plants, water will cross into the plant, but plants have cell walls here to give them structure, and they will not burst like red blood cells will. So water can actually swell the cells, water being hypotonic to the cells themselves, and the water will swell and keep the plant nice, give nice full structure. If the plant runs out of water, and the solvent around the cells, because there's not enough water in the plant, tends to be very, very hypertonic, the water will leave the plant cells and it'll lose its integrity. The cell wall will still be here, but the cell structure inside, which is holding the cell wall in place, will collapse. And so that cell wall will also collapse a little bit and lose its integrity, and the plant will wilt. So this is because of hypertonic solvent conditions around the plant cells. They don't have enough water. All right. So now this brings up the issue of how do we move things across membranes? If things can't cross membranes easily on their own, you need some way of moving things, right? So this is called transport and membrane transport. And there's different ways of doing this. If you have a difference in solvent concentration, solute concentrations across a membrane, so let's say this is a lot of, let's say it's glucose, like that 6-carbon sugar, right? On one side of a membrane, and not very much glucose on the other side of the membrane, right? There's a concentration difference. It's hypertonic in glucose to this, right? Well, the glucose is going to tend to want to move down its concentration gradient to equilibrate concentration on both sides of this membrane, right? So well, how do you do this? If you have just a membrane, the glucose can't pass, and it will, the concentration difference will remain constant, right? However, if you have a protein that allows glucose to pass through, then you can equilibrate the concentration of glucose. The glucose can move from outside the cell to inside the cell. This might be very useful for a cell who's using glucose to metabolize and is in an environment where there's a lot of glucose coming in the extracellular fluid that the cell's going to use. Like, for example, this might be blood, right? And this might be a cell that's bringing in glucose to use in metabolism. Blood's moving glucose through past the cells, and then the cells bringing glucose in. So these are called channels. They're just proteins that allow certain solutes to pass through. In this case, glucose. It can be for sodium, they can be for protons, it can be all sorts of things. Anything that the cell may need to move across its membrane. All right, channels are just simply a way a protein structure allows this thing to move through passively, right? Other ways of moving things through involve protein movement or rearrangement of protein shapes. So let's say this molecule here, this gray blob, whatever that is going to move across the membrane. Well, a carrier can allow that to happen sometimes too. The gray blob interacts with the protein rearranges its shape now so that the gray blob is ejected out the other side of the membrane. And that is a carrier. This doesn't require energy because the concentration of the gray blobs on this side are higher than on this side. So diffusion is actually powering this, but you do need the movement of the protein, the rearrangement of the protein molecule to do this. So this protein is actually flexible and moves a little bit to allow this to happen. Okay, so when you have a situation where you have a concentration difference between between two sides of a membrane, then channels and carriers will allow molecules to move down their concentration gradient. But what happens if you want to move something against its concentration gradient to establish difference in concentration, which cells do a lot of? You need some sort of way of doing this. This is called active transport, right? So active transport uses ATP here to change protein shape so that it can pump ions or pump something across a membrane to generate a concentration gradient difference. So here's sodium's indicated as the little hexagons and potassium as the little ovals, right? And what this protein here is a sodium potassium pump. What it does is move sodium from low concentration on the inside of the cell, in the cytoplasm that's called, to the outside of the cell, or extracellular space, this way. And if you notice, the sodium's at high concentration here and low concentration here, right? So sodium is going to move against its concentration gradient. And at the same time, this protein's also going to pump potassium from low concentration on the outside of the cell, high concentration on the inside of the cell. Now to do this, this is not an energy favorable process because you're moving things against a concentration gradient, which is against the energy state of these molecules. So you need to power this with ATP. So ATP is going to come in here, interact with this protein and cause it to change shape so that it will move sodium out, 3 sodium ions out for every 2 potassium ions it brings in. And it burns an ATP. It burns in the sense that it hydrolyzes ATP to ADP, lower energy state molecule. This is the higher energy state molecule. And that interaction, including dropping off of a phosphate in interaction with the protein, allows it to pump. So sodium, it loads the molecule here on the left, right? And then ATP interacts and that changes the shape of the protein so that the sodium gets diffuses out this way, and then potassium moves in to the new shape of the protein, and then this ATP hydrolyzes to phosphate and ADP, and that changes the shape again so that now the potassium moves out, diffuses out, the phosphate leaves, and the protein's back to its original shape and go back to starting this process again. So that's a sodium potassium pump. If you want to see this, this is actually a really nice video showing the function of sodium potassium pump in a molecular scale, but in a way that's understandable in this video. So I would suggest you go look at this. Okay, then there's this idea of co-transport where you can actually move things across a concentration gradient by using other things that are moving down their concentration gradient. So I'm going to show you an example in a second of proton concentration gradient being used to do this. But this is an example of glucose using sodium as a concentration gradient. So sodium on the outside of the cell is high concentration, as I showed you just now, and on the inside of the cell is low concentration. What the cell's going to do, and then the glucose, in this case, is high concentration inside the cell. What the cell's trying to do is bring more glucose in there, maintain that high concentration. So what we're going to have is called a co-transporter, which will bring sodium, move the sodium down its concentration gradient, and also bring glucose, which is low concentration, in at the same time. So it comes together with the diffusion. This doesn't require ATP because the sodium is moving down its concentration gradient. But it's still energy use because the concentration difference between the sodium on the outside and the inside is high. So the natural energetic state is to have the sodium move down its concentration gradient. So you're coupling this diffusion of sodium with diffusion of glucose against its concentration gradient. That's called co-transport. Here's another example. This is called proton coupling, where what we have is ATP being burned, hydrolyzed to prompt protons against their concentration gradients, active transport, right? And then coupling that with another molecule called a sucrose H+ co-transporter, which will take sucrose and will move it from low concentration to higher concentration inside the cell by coupling it with the movement of protons down their concentration gradient. So down they go, down their concentration gradient, and the sucrose goes with it against its concentration gradient. So this is considered active transport because you need to use energy to generate the proton gradient in the first place. Okay, so there's an overview of the transport mechanisms. Passive transport does not require energy. It uses the energy already stored in the concentrations difference separating these molecules. So here's diffusion down concentration gradient of something across the membrane. Here's diffusion of something that can't cross the membrane, but it's moving down a channel. So there's a channel just moving directly through, and then a carrier would move something else and involve movement of the protein that's allowing this transport to the other side. This is all called facilitated diffusion, by the way. Facilitation means something that's moderating something. Diffusion is moving of stuff across from high to low concentration, right? So facilitated diffusion, proteins moderating this diffusion. Active transport, using energy to move things against their concentration gradient. Okay, so now I would suggest that you look at the rest of these slides and look at some of these structures and what they're doing. They're pretty interesting. Maybe I'll make a separate video about this, just showing their various membrane crossing proteins and their functions. All right, I'll do that separately. All right, that's enough for this. Let's stop this.